All right, welcome back, team. I still can't get the webcam to work. It keeps lagging, so you just get my voice and these animations. All right, when we left off, we were talking about the life cycle of a population, and we'd also established the idea of a gene pool. And the last thing, or one of the last things I told you, was that we refer to the frequencies of our two alleles using the terms P and Q, and we call P the frequency of the dominant allele, so like in lab, the red beads, and Q is the frequency of the recessive allele, so in lab, those were the white beads. And as frequencies, these are numbers between 0 and 1, where 0 means that allele is not present, and 1 means that allele is the only allele. I have a slide on this later, but I might as well say it now. We are oversimplifying the world here, as we often do in an introductory class. Most genes have more than 2 alleles, but we're going to pretend like there are just 2 alleles. All of the math, all of the calculations we'll do would work for a system with multiple alleles. You just have to add some modifications. So we're looking at the simplest case. No surprise, life is more complicated than what we're doing today. Okay, so here's our life cycle. What we're doing with population genetics is saying that we have this adult population. We are observing them. We could observe their genotypes, their phenotypes, and we're asking ourselves, did this population come about through a process with evolutionary pressures or not? Okay, so what we're doing is taking our adult population and using it to calculate our expectation for what that population would look like if there was no evolution affecting it. Okay, so if there's no evolution, what this population looks like is the same as what its prior generation looked like, and it's the same as what its future generation will look like. So you can think about it either way. Like, we're calculating what the previous generation looked like, what the future generation would look like, and then comparing those calculations to what we see. All right, so we are coming up with a prediction about what we think our population would look like, or what we know our population would look like if there was no evolution. And then we're asking ourselves, is that actually what our population looks like? Okay, so just to remind ourselves about the life cycle. Adults make gametes through meiosis, and my animations aren't coming up. We'll get there. There we go. Meiosis. Gametes find each other randomly if there's no evolution. Randomly, they form zygotes. Our gametes with one allele for every gene come together and give us zygotes, individuals with 2 alleles for every gene. If there's nothing interfering with development, these zygotes grow into juveniles, juveniles grow into adults. So if we want to determine whether a population is evolving, we need to know some things. One thing you'll be given, and then the rest of the things we have to calculate. So you're going to be given some information about the population. We're going to start like you're given the number of individuals with each genotype. And we're going to just look at 3 genotypes: homozygous dominant, heterozygous, and homozygous recessive. Then you'll need to calculate the actual allele frequencies. So based on what we're given about the genotype frequencies, we can easily calculate the allele frequencies. We'll look at how we do that. The next thing that we'll look at is calculating the genotype frequencies that we expect based on the allele frequencies that we have. So given our gene pool, given our bucket of red and white beads, how do we expect these beads to come together into diploid adults if there are no other factors influencing this population, if there's no evolution influencing this population? How do we expect alleles to sort themselves out into individuals? Okay, so we'll get an expected set of genotype frequencies. And then the third crucial step is to compare our expected genotype frequencies, the things that came from step 2, compare those to our actual genotype frequencies that we were given. Okay. Oh no. Sorry. Okay. So here's an outline on top of the life cycle. You're given some information about the population, the number of individuals with each genotype. We calculate the allele frequencies. We'll get there. We'll do this all together. This gives us, this allows us to calculate our expected genotype frequencies, and we can compare those. So your mantra for population genetics is calculate, calculate, compare. And it's this compare step that's really crucial. A lot of students that struggle with this concept forget about the comparison, and they calculate things and then they sort of say, oh, well, this all added up and my formula makes sense, so I'm done. But that's not the case. We have to compare the things that we calculated to the information we were given. Okay, so our 3-step process. We're going to break it down. Here's that slide I told you about already. We are assuming that we're using a diploid organism where each individual has 2 copies of every gene, potentially 2 alleles, and every gene only has 2 possible alleles. So that's an oversimplification, but we're doing it. Okay, so we're given information like this. We've got the number of adults, the 3 genotypes, and here are their distributions. The first thing that we want to do if we're given numbers of genotypes is to convert those numbers into frequencies. This is important because frequencies can then be transferred to any population of any size. Numbers, 16 individuals, means something different if you've got 100 individuals than it does if you've got 1,000. So numbers aren't really useful outside of their own context. Frequencies can be useful in any context. So this math should make sense to us. We're calculating our genotype frequencies, and that's just going to be the number of individuals with the genotype we're asking about divided by the total number of individuals in the population. So because I got to decide the numbers for this really clear example, we have 100 individuals in our population. If 36 of them are homozygous dominant, that's 0.36. Or you could think about this like a percent, 36%. Okay, 48 individuals have the heterozygous genotype, so 0.48 is that quantity. And then what's left, 0.16, is the frequency of the homozygous recessive genotype. And you can see that we have this shorthand here for genotype frequency. We're busy scientists. We don't want to write genotype frequency out all the time. So it's a big G and then a subscript with the alleles or with the genotype. So big G and then a subscript, big A, says the genotype frequency of the homozygous dominant type. Okay, so genotype frequencies. You'll notice that just like if we add up the numbers of each individual, that adds up to the total number. If we add up their proportions or their frequencies, that adds up to 1, and that 1 means 100%. Okay, so we're really used to math where we're kind of like adding things up, and then whatever's on the other side of the equal sign is some kind of important answer. Here, that's not the case. The fact that these 3 things equal 1 just tells us that we've done our math correctly. If we have all 3 types, we've got the proportions of all 3 types, they should equal 100% of the types. Okay, so the fact that this equals 1 is no revelation, it just reinforces that we've done our math correctly. All right, so we just did going from genotype numbers to genotype frequencies, and this is going to let us go convert to allele frequencies with some simplicity. a side note, we can only literally count the genotype frequencies if we can see the difference between homozygous individuals, homozygous dominant individuals, and heterozygous individuals. This is not something we have talked a lot about, but you might recall from other studies you've done that sometimes the heterozygotes look different from either of the other two genotypes. And in the examples we've done, sometimes they don't. Okay, so you can't always look at individuals and tell their genotype. Okay, so we calculated our genotype frequencies. The next thing to do is use those to calculate, our allele frequencies. So what does our gene pool look like? You could think about this as going from adults to gametes, from adults to the gametes that they contribute. Okay, so we have to take our individual genotypes apart into their alleles. So we're going to look at this the longhand way, and then I'll show you a shorthand way. And you can do whichever one you like. All right, so we could think of our 36 individuals with two big A's, two dominant alleles. 36 times 2 is 72, so there's 72 big A alleles from those individuals in our gene pool. We've got our 48 heterozygotes, and they have 48 of each allele, so we've got 72 plus 48 big A alleles. Then we've got our 16 individuals that each have 2 little a's, so that's 32 little a's from them. So 72 plus 48 is 120 big A alleles. 48 plus 32 is 80 little A alleles. And we can also sum down the columns just to make sure we've got everything right. If we've got 100 individuals, if they're diploid, we should have 200 alleles. And we can also add 120 with 80, and that's 200. So we've got everything accounted for. All right, so just like we went from number of individuals to frequencies of individuals, we really want to go from numbers of alleles to frequencies of alleles. Because it's possible that each individual contributes more than 2 gametes. It's possible that all individuals contribute more than 2 gametes. So number of alleles isn't transferable outside of its context. We want to go with frequencies of alleles, which we can then use in any context, in any size population. Okay, so that's our long-handed way. This way should also make sense. This is what we did for genotype frequencies, where we have P equal to, so first we have to do our long-handed way, and then we can calculate these proportions. So number of big A alleles divided by the total number of alleles, 120 divided by 200 is 0.6 or 60%. Okay, there's the same calculation for Q. You'll notice that 0.6 and 0.4 add up to 1. We'll come back to that we've got 100% of the alleles accounted for. Let's look at the shorthand way. So if we already have our genotype frequencies calculated, we can calculate allele frequencies straight from that. And here is the formula. The frequency of the big A allele, which is P, is equal to G big A, the frequency of our homozygous genotype, homozygous dominant genotype, plus half the frequency of big A little a, our heterozygous genotype. So there's that, and you see that math works. The frequency of our little a allele, also known as Q, the same, the frequency of the little a genotype plus half the frequency of heterozygotes. Okay, and this comes out also to 0.4 like we saw before. Okay, and you'll see that P and Q always equal 1. This means that we've accounted for all the alleles. If we take up half of the alleles, or one type of the allele, add them to the frequency of the only other type of allele, we've got 100% of the alleles. Okay, so this doesn't mean anything in terms of evolution, just means you've done your math right. Okay, so next we're going to go to zygotes, but before that you've got a little assessment. Here's what's on it. take that and then come back for the third video. Again, no grade here, just participate.